Intro to Bio class, our final assignment is to create a video where we teach a topic of our preference that we previously learned in the semester. For this final assignment, I decided to explore the concepts of cellular respiration with a concentration in meiosis. As we all know, every organism is a result of cell division. The ability to procreate is one of the characteristics that best distinguish living things from non-living matter. When a cell goes under cell division, the two daughter cells that result are genetically identical to each other and to the original parent cell. Cell division is vital for many organisms. We have damaged cells that have to be replaced with the millions of cells that divide every second. Reproduction is another reason. Single-celled organisms reproduce by dividing in half. Sexual reproduction cannot occur without the fertilization of an egg by a sperm. In this case, meiosis is involved, and I'll explain how in a few seconds. The cell cycle is a series of events that take place in a cell as it grows and divides. The tetrad is a set of 2 chromosomes. Each chromosome will meet its counterpart. 2 of the chromatids of those chromosomes are going to be joined by points called synapses. Those points are the ones that are going to recombine, and when the chromosomes are separated, we're going to obtain new genetic combinations. All this happens in prophase 1. Prophase 1, there are several substages, but the most important thing we can remember about this stage is that each chromosome meets its pair, and that occurs in recombination. In metaphase 1, what is going to happen is that the pairs of homologs are placed forming the equatorial plate. That is to say that they are arranged in single file in the middle of the cell. It will be the homologous pairs, not the individual chromosomes. When the homologous chromosomes reach the anaphase, the centrals are going to stretch from the homologous chromosomes, and we're going to end up with one chromosome at one pole and another chromosome at the end of the other pole. In telophase, what's going to happen is that the chromatin should begin to decondense, and the nuclear envelope should reappear with the last step, cytokinesis. That is division of the cytoplasm. Another characteristic of meiosis is that this happens kind of slow compared with mitosis. Mitosis is much more easier, while meiosis is spliced with the second division. In division 2, we go directly to prophase. Interphase doesn't occur. We want the cell to become haploid daughter cells. Division 2 looks a lot like a meiotic division. In prophase 2, what we find in each of the daughter cells is a chromosome. Therefore, in that prophase 2, the nucleus is undone again, and the chromatin, if it had relaxed at the end of division 1, is condensed again. In anaphase 2, the centromeres of the sister chromatids separate, and the sister chromatids of each pair move toward opposite pole of the cell. Telophase 2, nuclei forms at the cell poles, and cytokinesis occurs at the same time. There are now 4 haploid daughter cells, each with single chromosome. And that's all with the process of meiosis. I hope it was easy to understand, and thank you very much for watching this video.